Next thing we have facilitated. If you're taking notes, maybe make an arrow and say facilitated means help. It needs help. It needs help for the molecules to pass through. It just can't happen on its own. The help is through this transport protein. So facilitated, it means help. It needs help for the things to move across. Facilitated diffusion is passive. There's no energy needed still, as long as the molecules are moving down the concentration gradient. So facilitated diffusion, because we said diffusion definition was things are moving from high concentration to low concentration. It's still the same. It's moving from high concentration to low, but maybe these have a charge or they're too big in size. So they need a channel. So they're gonna go through channels. If you use the sodium, so you guys have a everybody has a sodium potassium pump in the muscles. So since we know those are hydrophilic and these tails are hydrophobic, it doesn't just go across. It's gonna need a channel to go across. So it's gonna need a channel. It's gonna need help to go across. And it's going from high concentration to low concentration. So substance move across membrane with assistance, with help of transport proteins. So we covered diffusion, osmosis, and facilitated diffusion. Those are passive, means they don't need energy. So this is an example I was giving you. Hydrophobic tails of phospholipids repel hydrophilic substances. They're water-faring, they don't like water. And sodium chloride that are hydrophilic, they need to pass through a channel to cross the membrane. They just can't go across because of these tails that don't like water, basically. Now we have active transport. Now I want you to look at the picture and tell me what's different in this photo. So in this photo versus this photo.
The energy, ATP.
There's energy. So active, you're gonna need energy. What else? So you have this photo and then you have this photo here.
It's inverse for the concentration moving.
It's going against the concentration gradient. It's not going from high to low anymore. It's going from low to high. This is going against the concentration gradient. This goes back to, again, when we want to keep high number of balls in one area versus low in the other area, that's going to require energy. Just going against the concentration gradient, it's going to need a protein channel and it's going to need energy. So it needs both. ATP breaks down into ADP, releases energy. It's going to cause these balls to jump to the side. When you go to the dentist, they give you anesthetics. So you have sodium potassium pump. It's going to basically block those potassium channels and it's gonna stop this whole process from happening. And it's gonna, basically, you're not gonna feel anything. Your nervous system kind of stops working in that area if the channels are blocked. This is sodium potassium pump. I'm not gonna go in detail for this. This is anatomy. They will cover it for you. So in this photo, all it's trying to show, there are a lot of these purple balls outside. There are few purple balls here inside, but still it wants to move outside. These want to go outside, even though it's lower concentration here. For that, we do need ATP. ATP turns into ADP, releases a phosphate, attaches to the channel, causes it to open, and the balls can move outside. This is how sodium potassium pump works. Your muscle movement, your muscle contraction. It's called sliding filament theory. You guys don't have to know that, but it's in anatomy. So this is what they block. They block these sodium channels or potassium channels actually when they're trying to stop your nervous system from working. They just block these channels. Next thing we have is endocytosis. Endo means inside. We learned about endomembrane. Cyte, cell. Exo means out. Tosis is the process. This is called endocytosis. It's going to allow the fluids and large molecules to move inside the cell. Let's say this is an amoeba that's swimming at the bottom of a pond and it sees some food. Once it sees some food, it's gonna start drawing its plasma membrane inward. And eventually it's going to turn into a vesicle and it's gonna bring that food inside. So engulfing things inside is endocytosis. Any questions on anything so far? Everything's making sense? Yeah. Still go back and review it before the exam. Next one is exocytosis. Now, some of the waste materials need to go outside. It's going to be packed in a vesicle. Maybe lysosome broke it down. Now it's packed in a vesicle and needs to go outside. It's gonna start fusing. It's gonna start fusing with the membrane like this. And in the end, it's just gonna expel the contents outside and the membrane just fuses with this membrane. So membrane of the vesicle fuses with this membrane and the contents are spilled outside. That's exocytosis. Uses vesicles to transport substances out of cells.